start off by saying a big thank you to the entire committee for organizing everything and for all of you for being here. Also, this is a project that's really dear to my heart, but it is really still ongoing and there's a lot of moving parts. So this is really preliminary data, so bear with me. This is also the first time I've actually tried presenting any of it, so we'll see how it goes. But I am really happy to show you. Let me know if anything is unclear or if I start talking too fast, because I sometimes do. Okay, doesn't work. Yeah, it works really well. Comment je fais pour passer? Ah, okay. All right. Thank you. Okay, so just a quick recap for anybody who's not a biologist or who's kind of forgotten, but predation pressure is a super important natural selection. It not only affects adaptive traits, but it also impacts interactions between organisms and their environment. It can affect diversification and speciation. So it is very important to study, and it affects all organisms. And organisms can use different strategies to avoid being attacked by predators. And they can range from avoiding predators. So this is like crypsis or camouflage, hiding from predators so that they don't get attacked. It can also go to the other direction completely and deter predators from attacking. So if you're chemically defended, or if you have physical defenses like spines or hard shells, then you're protected from predators that may try to attack you. In the case of chemical compounds, organisms that are toxic, they are also often combining this with signals to advertise their unpalatability. This is called aposematism. So this is quite common, for example, in certain frogs or certain butterflies. They're brightly colored to advertise to their predators that they're toxic. Don't eat me, you'll regret it. And this is something that's often studied. Aposematism is a common theme of study. However, it is generally studied for color pattern, mainly because we're visual. And so it is easy to see there are differences in color pattern. And so we are ourselves tend to study that particular trait. But chemical compounds or defenses that are present are also a trait that could vary in the same way as color pattern and could therefore be extremely interesting to study in determining how they vary and how they impact these organisms. So my particular model are butterflies. They're called Ethomini. They're often called clear-wing butterflies. They're exclusively neotropical. They're highly diverse. They're all toxic. They're brightly colored to advertise this toxicity. But a lot of species also have some transparency to some degree. But this transparency isn't, they're not fully transparent. They'll have these colorful elements. And so these are thought to be signals to advertise their toxicity despite their transparency. So they're widespread in the neotropics. They're quite abundant. They dominate mimicry rings. So I don't know if you can tell, well, you probably can't tell, but these are two different species of Ethomini. They're just mimetic. They resemble one another. Same thing here. Actually, this is Ethomini, a Melanea, and this is actually a Heliconius. So they're mimetic, but they're actually not closely related, but they're both toxic. So these are different mimicry rings that exist. Usually what happens is predators will purify a signal. So you'll find in a certain habitat, you'll find one particular mimicry ring that dominates the habitat. So as I mentioned, I'm interested in the toxicity, and Ethomini are particularly interesting for the toxicity, at least as far as I'm concerned. So the basal genera actually feed on Apocynaceae, which actually contain a type of alkaloid that are called pyrrolizidine alkaloid. I will not pronounce that for the rest of the talk. I'll just say PAs. So those are the basal genera of Ethomini. They get these toxins from their plant. Meanwhile, though, most of the ethomini have actually shifted host plants to the Solanaceae, which are nightshades. So plants like tomatoes, eggplants, things like that. So they're also toxic. They contain alkaloids that are different from the PAs, but they're also quite toxic. And we believe the butterflies to also sequester those compounds. And this here is just a figure of showing the different Solanaceae, the different ethomini. And basically the ethomini tribe has shifted to basically utilize all the different Solanaceae. They're quite varied in their use. Basically, you'll have generally a genera that is specialized on a genera of the Solanaceae. But for the tribe in total, we basically have a whole lot of different host plants. What is interesting is that the adults of those butterflies that use the Solanaceae as host plants still depend on the PAs. And so they will collect these alkaloids from other plants once they're butterflies. And this is interesting. It means that there's something that is still pressing selection on them, putting selection on them to continue collecting these compounds, despite the fact that they have access to other chemical compounds from their host plant. So what does this mean for the toxicity of these butterflies? Like what factors may be correlated with it or may impact it? Well, like I said, resource availability. So the different host plants they utilize or the PA sources they utilize in the habitat. The other thing that may affect it is predators. Like I said, aposematism is a defense against predators, and therefore we assume that in habitats where there's increased predation pressure, we'll also see either higher toxicity or higher conspicuousness. These traits should vary with predation pressure. The types of predators also may impact. So basically, if we have invertebrate predators, they may act differently to avian predators. And of course, conspicuousness. Like I said, some of them are somewhat transparent, other are fully colored. So depending on how conspicuous the signal is, it's considered to be an honest signal. They're not cheating predators. They're definitely advertising that they're toxic. So if we have a more conspicuous signal, we could assume that possibly they're also more toxic to those predators. And microhabitat can vary directly or indirectly with this. So this project is actually ongoing both in French Guiana, in Peru. We're sampling 6 sites per country. Basically, we have 3 classes of altitude, 2 sites per type of altitude. So obviously, you'll notice here that there's only 5 points, and that's because the 6th point still hasn't been sampled. We're hoping to do that in the next few weeks. But we already have in French Guiana over 500 butterflies collected. We've also collected the plants that we find that are potentially host plants or PA sources. So yeah. So real quick, we looked at site discrimination as a function of mimicry rings and the species that are present in the different sites to see if there's any assemblages that are more dependent on certain types of altitude. And what we find is similar for both the mimicry rings and the species. So I'll just stick to one. Basically, sites actually cluster by altitude. So different altitudes have a tendency of having different types of species and also different mimetic communities. The higher altitudes are also they have more abundant species and mimicry rings and more diverse ones. And this doesn't seem to be necessarily linked to those butterflies that are conspicuous. The mimicry rings that are conspicuous, that are brightly colored, tend to be present at all the different altitudes, although the species assemblages that are present may differ between the different altitudes. So, one species may be replaced between the low and the high altitude, but they'll have the same mimetic pattern, is basically what I'm saying. However, what we do see is that the butterflies that are more fully transparent are actually exclusively at higher altitudes and intermediate altitudes. They're absent from lower altitude. So that might explain why the abundance and the diversity is higher at higher altitudes. So we've started looking at what could be explaining this. So we have looked at some abiotic or environmental factors. There are some differences, at least for the really higher altitude, have lower rainfall and lower temperatures, which makes sense. Not sure exactly how that will fit with explaining the more abundant, the abundance and the diversity that we see at the higher altitudes. But one thing that we're currently looking at, we do have the data, is the canopy cover and the light intensity. And the reason why this might be important and interesting is, well, these butterflies, if there are more butterflies that are transparent at higher altitude, transparency, lower conspicuousness probably has something to do with detection by predators. And so it makes sense that certain environmental conditions like these would impact how the signal is received by the predators. So these are factors that I'm really looking forward to checking out. Similarly, we do know that transparent species are less conspicuous because we did test it. So real quick, because it's a lot of data, I'm aware of it. But this is the result of modeling predator vision. So we use the blue tit. I'm aware we don't have blue tits here. Unfortunately, we don't have the data, the physiological data for the butterfly predators that are present locally, but it still transfers. Like the trends we find will still work. And what I want you to see right now is like, this is increasing distance here. The red line that you see is the limit, they shouldn't have split the word in two, but limit of detectability by the predator. So basically the red line shows you above it, it detects it, below it, it's a lot less likely to be able to detect it. So the more a butterfly is transparent compared to the completely conspicuous one, then the more likely or the less likely it is to be detected, especially at further distances. So basically transparent species are less detectable and even more so when they're far away. So that could be something about the habitat, the amount of light there is in the habitat and stuff like that. It could also be a question of, you know, how much predators there are, like what are the predator communities in these different sites. I've just said that, but so we did look at predation pressure, and the way we do that is by placing, these are artificial butterfly models. So they're real butterflies that exist in these habitats. They're not toxic. They're palatable by birds. And we placed 200 of these artificial butterflies at each site. So you can see that, yeah, obviously I'm still missing a site. It's ongoing. One thing that I'd like to point out is the predation rate is super low. It's between 1% and 3%. This is unusual. It shouldn't, I've never seen it that low. In Peru, it's actually quite a bit higher. It's more than twice that. But one thing that is similar to Peru is there seems to be more differences between the sites than there really are between the altitudes. This still needs to be tested, obviously, because depending on what we get for our third site, well, our second site at low altitude, maybe we'll find a difference with the higher and intermediate altitudes. But it looks like there's no real difference to explain why some butterflies are less conspicuous at higher altitudes, at least for predation pressure per se. So maybe it's not the predation pressure per se, but maybe it's the types of predators, like maybe toxicity is different for transparent or less conspicuous butterflies, and that has an effect on different predators in a different way. And so real quick, because it's a bit of a mouthful, this experiment. Basically what we did is we tested like a dozen butterfly species with chicks. And basically what we do is we present them, we crush the butterflies in chick feed, and we present them to the chicks with different colors. And then we measure how quickly it takes them to learn to avoid the chick feed that has the butterfly. So these will be averages of how quickly they learn. So the higher the value is actually means that they haven't learned, they keep eating them. And then lower values means that the butterfly is probably really toxic and they learn really quickly to avoid it as much as possible. We also did that with spiders. Basically, we put in, I think it's 20, tested 20 per individuals per spider. We put in a butterfly and we see if it's rejected by the spider or if it's killed by the spider. And this here is mortality rate. So the higher the mortality rate, the less likely the butterfly is very toxic. And we tested this with a mix of very conspicuous butterflies and less conspicuous ones. And basically what we find is that it doesn't really matter how conspicuous they are. It doesn't seem to affect toxicity. Transparents are just as toxic as conspicuous ones. And to top it all off, those that are really toxic to birds aren't necessarily the ones that are most toxic to spiders or vice versa. So like this one here is clearly pretty palatable to birds. It means the birds don't reject them really, but they have an okay survival rate, from spiders. So clearly, either different predators respond differently to the compounds or they respond to different compounds. So we haven't done all the chemistry yet. That's the main thing to take away. But we have some preliminary data on PA content. So those are the plants that they visit as adults. And so when we look at, again, the speed of aversion learning, so here, the speed of aversion learning of chicks. So again, this is the most toxic, this is the least toxic, and this is PA concentration. And the same for the spiders. So most toxic to least toxic with PA concentration. And basically, there's no clear relationship between PA content and how likely they are to be rejected by different predators. So I'm not saying that the PAs don't have a role against predators, but it's clearly not straightforward. So we started looking into this, and this is like data from like barely a few weeks ago, which is why I don't have like the measured data specifically for aversion learning and stuff like that yet. Those are the nitty-gritty data. So these are the averages of the 6 chicks. They're tested 12 times because we look at not only how quickly they learn aversion, but how long it lasts. But I'm just going to focus on the first part of the graph. And what I want you to look at here is, okay, so if we have some butterflies that we reared on a single host plant, and then some of them we gave them access to PAs, and those we didn't. So the ones that do have the PAs are in dashed line. And what we find is that in a lot of cases, adding the PA, giving them access to PAs, does provide some defenses against predators like chicks, so bird predators. So PAs clearly have some kind of role in defenses against avian predators, although we saw earlier that it's not like a direct relationship between the two. So we also looked at host plants. So the different colored lines are actually host plants. And if we look at those, in a lot of cases, there are differences between different host plants. So if you give them one host plant, they'll actually be less toxic than another. In some cases, they're just as toxic regardless of what host plant they get. But it gives us some kind of window into understanding these euthymini. Basically, they seem to have these 2 different types of compounds that they sequester and that they utilize, and it seems to interact together, although we don't really know exactly how at the moment. And I'm already at 20 minutes, so maybe I'll skip these and just give you a brief overview. Just remember that at butterflies of the euthymini tribe are more abundant and more diverse at higher altitudes. This is likely the result of differences in abiotic factors, including canopy cover and light intensity, which we still want to look at, especially since these could impact the transparent butterflies that are more common at higher altitudes. Predation pressure doesn't seem to impact the communities themselves, although different predator types do seem to respond differently to the different compounds. And by affecting toxicity, resources could affect distribution and diversification in this tribe. This is still ongoing, so we are currently analyzing all the chemical compounds that are present in all the butterflies and in all the plants. So then we can link the two, but also link between the different taxa, the species that have that are closely related, or how they utilize their microhabitat. So we can determine the selective pressures for this and determine how it impacts distribution or assemblages or diversification. We're also looking at whether there is a relationship between defenses and mate attraction. So in organisms that are aposematic, often the visual signal changes depending on the geographical distribution, and this may cause diversification. And we don't know if this is also true for those compounds, even though we do know that in some cases those compounds that dictate toxicity, that dictate relationships with predators, are also the same compounds that may affect courtship. So just want to say thank you to everybody working on the project. I have collaborators, but also these are my 2 PhD students who didn't show up today. And so if you do find them, because now that you have their picture, you can track them down and ask them all the questions, okay? And I have a bunch of other students.
Any questions?
Just for curiosity, you work in which institution? Because you didn't put that up.
So my first slide, sorry.
I missed it then. Huh? Ah, you mentioned. Who? Huh? Liza, but you are in which city?
I'm sorry?
In which city?
I'm in Cayenne.
Ah, in Cayenne, okay, okay. And in Peru, you work in which altitude?
So, I don't know if you know Peru. I'm in
Yeah.
the Amazonian region called a city called Tarapoto, and I work at the same altitude classes that we have here. But basically, so it range the same range, but basically there's a city just above it that's called Guayabamba, and that's basically up to where I go. It's like basically a San Martin region, and then I go lower, really low altitude that's equivalent to here. Yeah.
So it's in the Amazon basin, obviously.
Exactly.
Yeah.
But also with higher altitudes. And the interesting thing is, well, because Peru is not as flat as California, there are a lot more mountains. We do get a lot more of this trend of really transparent butterflies at higher altitudes, and even those same altitudes as this.
That's what I was getting at, because in Peru you go much higher than here.
Yeah, clearly. But even without going higher, you still get more diversity, more abundance of these different butterflies.
And here we are not in the Amazon basin.
No, sure.
We are in another place. Thank you. No, I'm quite familiar. I am a botanist, but I'm familiar with this because one of my professors was Larry Gilbert. So I have seen a lot of heliconius and a lot of mimicry, all kind of things, mostly in Costa Rica.
Well, he's the grandfather of basically all my collaborators.
Yes. Okay. Thank you.
So the butterflies are only storing the PAs, or are they also modifying them?
So they're mostly sequestering them, and actually just so someone has the 80s done. I don't know. Do I need the microphone? Or yeah. Oh, sorry. Okay. I hate the microphone. But basically, some studies have been done in the 80s where they basically tracked how it was how it was sequestered, like how it was kept in the tissues without causing damage. And they show that they're able to just modify it enough during while it's in the body, but it converts to its original form when it's attacked, and then it becomes toxic again. So it's transformed just barely so that it's not too toxic to the butterfly while it's stored in the tissues, but then it's released in its full form. So it's not a precursor to a toxin. It really is the toxins that they're sequestering, at least for the PAs. I don't know about the host plants. Okay. Because actually until now, the literature has always assumed that they're collecting these PAs because they're unable to sequester the alkaloids from the host plants. And we're finding that this is clearly not the case.
All right. Okay, thank you.
I have a question.
Yep.
Yeah, I have a question and a comment. What about seasonality in the sampling of the different sites? And because I understood it's a one-shot sampling you did, but are there butterflies and caterpillars all year long?
Yeah, so the Ithomines are present all year round. However, the problem is, it's easier to collect them in the dry season. In the rainy season, there's just fewer of them and they tend to be dispersed a lot more. So it just makes it a bit more complicated. I have data from Peru where I don't find a huge seasonality difference in the community assemblages. I was thinking of maybe repeating it in French Guiana, but I'm worried because the numbers of butterflies, although it's good, we have like 500 or whatever, it's actually quite low compared to other places. So I'm not sure if I'm going to redo it. I was thinking of doing the predation again, but again, I'm still worried. I don't know if it's lower than the dry season. I'm hoping it would be higher, but if it's lower, we won't see a difference. So I'm still on the fence. If you see me out in the field in winter in the middle of rain, it's because I've decided to do the predation at least. So we'll see.
Okay, And I have a comment.
Yeah.
Then why not? Why your title is the chemistry of war and not the chemistry of peace, which would be the same, would mean the same, but in a different way.
I don't know if we could say it's peace though, because I mean, they're not brokering peace, at least not for the toxicity. Maybe the signal could be brokering
If
peace.
you have a good defense, you are not attacked. So
That's true. That's true. But they are, you know, they could, we don't know how much they're attacked though. So I don't know.
Thank you.
Oh.
Yeah. Oh, yeah. Sorry.
The last hypothesis or question that you raised regarding the relation between mating and defense,
Yeah.
I found that very interesting, and I wondered, because I've never thought about it actually, if this is a thing in other insect families, or how did you come to this question?
So it's not directly that way, but in the literature, there's plenty of examples of different organisms, especially insects, you know, when they change host plants, they tend to change their pheromones, and that causes reproductive isolation, and it can cause diversification and speciation. And we do know for aposematic organisms that the visual signal, so the color pattern, this happens a lot. It's called a magic trait, basically, because it affects not only natural selection, but sexual selection as well at the same time. It's the same trait that has two impacts. And so my idea is, okay, well, if these butterflies are changing host plants and it changes their chemistry, or the resources they use changes their chemistry, then there's no reason why those same compounds can't impact both predators and their potential mates. And that could also cause diversification. And I haven't shown it here, but I do have a little bit of preliminary data where I don't know about for the reproductive isolation yet, but at least some of the compounds do change depending on the host plants or the PA sources. So at least sequestering PA compounds does change their pheromones. So it is a signal of something. So yeah, but it's really still ongoing.
Thanks a lot, Mélanie.